Hello, my name is Jonathan Ng. I'm currently a P3 student of Roseman University, and today I'm here to present my research. I am currently interested in a residency that emphasizes in emergency medicine, critical care, or trauma, and I would like to present my research today on testing C. elegans as an experimental model for dopamine agonist effectiveness in Parkinson's disease. As many people know, Parkinson's disease is known for the overall dysregulation of dopamine that happens in a human being during middle to late age. Symptoms of which can be associated with that of tremors, bradykinesia, meaning that motor functions are off, along with that of inability to walk or have strong gait. This is why one of the current methods of treating this disease has been dopamine supplementation through the usage of levodopa or carbidopa. One of the reasons of why it has been shown to have been efficacious in that matter. It has been correlated with as well GRKs, also known as G protein coupled receptor kinases, which have been known for, which by activating these receptors by the dopamine supplementation has been able to slow these symptoms down. My emphasis of research today has been put into the usage of dopamine agonists, meaning medications that are designed to not supplement the dopamine inside of the human system, but to activate the cells and that way it creates cascades without actually having the dopamine for patients that are contraindicated. As typically, dopamine agonists are second line in comparison to giving dopamine supplementation, which would be considered first line. There are three kinds of these GRKs in humans, known as GRK1, GRK2, and GRK3s. Now, the question that I might be given, or I'm probably going to have asked, is why C. elegans? Why these little roundworms that I find in the dirt whenever digging around inside of the garden? And the answer is because C. elegans are a model organism. The C. elegans has 41 to 59% genetic similarity when comparing humans to that of the C. elegans. So, in other words, that many human diseases can be observed in C. elegans, just like Drosophila fruit flies. But unlike Drosophila fruit flies, the dopamine receptors within the C. elegans is far more human-like in comparison to that of Drosophila fruit flies, ergo my decision to use these organisms compared to that of those organisms. There are two different kinds of GRKs that have been seen in C. elegans, known as GRK1 and GRK2, which are similar, if not completely alike, in their design and structure, therefore being the subject of my research here. The way that my experiment was set up during this time was I compared the egg-laying behavior when I treated these worms in the separate petri dishes with that of different dopamine agonists. I looked at egg-laying behavior because the way that the C. elegans was able to lay its eggs is based upon its own dopamine regulation within its own body. I wanted to measure the activity in this matter compared to, say, movement, because of the fact that I wanted to make sure that my research was as least objective as possible. I wanted to make it as objective as possible, as seen from these graphs right here, so then I could be able to monitor and regulate the overall trends of the data of which were occurring compared to just interpreting and seeing and being conflicted about how much that a worm would be affected versus not. Then I would look at the 3-day-old C. elegans that were treated with different concentrations of the medications pergolide, bromocriptine, and cabergoline in different types, also as stated as wild types, GRK1 lacking, and GRK2 lacking. Dopamine receptor activation was measured by number of eggs laid in a specific time. I had a clicker, I was counting them under the optical microscope, so much fun. So then, as seen here by the effects of pergolide, it was interesting to note that while looking at the data, when looking at the egg-laying rates after one day, it was a seemingly constant peak and then tapering off effect, such as seen from here from wild type. It seemed that after getting past the first concentration, it would reach like a peak or concentration, and then it would taper off downward. And the same would go for the GRK1 lacking and GRK2 lacking. I attribute this to the mechanism of action of the pergolide, which was designed to be more mutually just affecting G1 and G2 receptors in general. And then when looking at bromocriptine here, for example, It was interesting to note as well in data, knowing that this one is mostly GRK1 mechanism of action centralized, that after one day it was significantly less in egg laying rates compared to the wild type and then the GRK2 lacking, where it would have, as seen from this data as well, a kind of effect where it would hit some kind of a peak and then it would taper off after that. And the wild types as here hit a kind of peak and then taper off after that. For both of the 1 hour ones, there wasn't really like any strong correlations, and I believe that to just be the amount of time that the worms were just sitting in this petri dish with not really as much time to be able to absorb the environment around them in order to be truly affected by said medications. Then, as seen from right here for cabergoline, this one was more it would more function on the GRK2 receptors, so it was more predictable to note that, as seen from bromocriptine, that there would be over less egg laying rates when looking at GRK2 compared to GRK1, though GRK1 did have that kind of reaching a peak concentration and then tapering off as beforehand, while the GRK2 just had a gradual increase overall, as seen from both sets of data from looking at after 1 hour and then comparing it to after 24 hours. So in summary, those were my results, as stated from here, where it was somewhat predictable to note the various trends in data that had occurred, but I had no idea how much of a time where it would be overwhelming the system of the C. elegans to the extent that they would produce as many eggs beforehand and then just more like taper off after that, said concentration. And then future directions that I would like for this research to go into would be to look into the data collection of these specific medications, just more of it. When I was doing the study, I did it over the course of 6 weeks. Therefore, I would like to see that it would be used for greater potential in that, just understand more about the mechanisms of action, how they just affect them overall, along with just more data, so I can get understand better the overall trends and behavior, and then more exploration into other dopamine agonists that haven't been studied here, just because from what has been studied from clinical standpoint, there are 5 total dopamine agonists that only go for Parkinson's. There are way more that we could look into that we just only know so much about just because we haven't really done as much research into it. And then I would also like to see continued research into the aging C. elegans in relation to human populations, such as the C. elegans were tested after 3 days, and knowing that the life cycle of a C. elegans is 4 to 6 weeks, it would be more helpful, I believe, if the C. elegans were around 3 or 4 weeks in age, just in that way, near the end of their life, for most people, that's when Parkinson's disease does kick in. Therefore, I would like to see in the future, we could do more research into cross-aging studies just to understand how much it would correlate. Along with how I would also like to see just overall understanding of this, just because, as seen from the data, there is prediction that with higher amounts of with the dopamine agonist concentration, there is effective behavior. Therefore, these medications do work, but I would just like to see overall how much do they work in comparison to that of the competitor. Thank you for your time. I hope that if you have any questions, please feel free to say any questions to me directly as a presenter through my contact information. And if everything's good, then have a great day.